Welcome everyone to the fourth webinar of the New Zealand Marine Science Society. My name is Marijke Baboeder and I will be chairing today's session. This webinar will be recorded and you will be able to find it online on our YouTube channel from tomorrow onwards. If you have any questions to our speakers during or after their talk, there's a Q&A button on the bottom of your screen, so feel free to leave your comments there. Today's session has the title Marine Mammals and Seabird Conservation. However, we had some last-minute changes, and it turned out that all our presentations will be around marine mammals. So I'm very sorry for all the bird lovers in the audience, but it will be awesome, I guarantee you. Our first speaker is Victoria Warren from University of Auckland. Victoria is a bioacoustician who recently submitted her PhD thesis. She specializes in marine mammals, and her thesis investigated the distribution of baleen whales around central New Zealand based on acoustic detections. Enjoy the talk.
Thank you very much. So I'm just going to share my screen now. Cool. So good afternoon, everyone. I'm Victoria, and I'm going to be presenting a chapter of my PhD thesis, which uses passive acoustic monitoring to discover more about blue whales around central New Zealand. So around New Zealand, there are two subspecies of blue whale that co-occur. There's the Antarctic blue whale and the pygmy blue whale, but it can be difficult to distinguish between these two subspecies visually. And because of this, both subspecies are currently considered data deficient in New Zealand, and that's in spite of the fact that Antarctic blue whales are actually critically endangered at the global level. So in order to improve our understanding of blue whale distributions in New Zealand, we were interested in a long-term monitoring strategy that covered a large area, so we considered using passive acoustic monitoring as a possible methodology. And actually it turns out that acoustic monitoring is definitely a good option to study blue whales. So the song calls that are produced by male blue whales differ significantly between the two subspecies, as demonstrated by these spectrograms. So a spectrogram is a visualization of sound. So along the x-axis, we've got time, along the y-axis is frequency, and then the color scale indicates how loud a certain frequency is at a certain time. So you can see that it's much easier to identify blue whale subspecies acoustically than visually. So building on that, in 2016, JASCO acoustic recorders were placed in 4 locations in the marine environment around central New Zealand. So the 2016 deployment are the black circles on the map here, so there's 4 of them. So the recorders were deployed by NIWA, left on the seabed for approximately 6 months where they continuously recorded sound, and then they were recovered via the use of acoustic releases and buoyancy aids. And the data were downloaded back on dry land. And actually the promotional picture that we used to advertise this talk is me sitting with the equipment that we used for this. And then in 2017, the recorders were redeployed at 3 sites for another 6 months in 2017, and that's the white triangles. So I conducted a manual analysis on the data to detect the blue whale song calls. And if you remember from the first slide I just showed you, you can see that this file contains some pygmy blue whale calls. So I'm just going to circle them for you here. So I analysed a subsample of all data from all stations and across both acoustic recording periods. So I took 1 file from every 2 hours of recording and annotated all the blue whale calls. And this gave me a random stratified subsample across all 4 recording locations and across both deployments. So from this, we could examine the temporal distribution of blue whale calls. But temporal distribution was only one aspect that we were interested in. We also wanted to look into the fine-scale spatial distributions of the blue whales, but that's a bit more tricky. So blue whale calls are really loud, so approximately 190 decibels, and as such they can travel long distances underwater, and especially in deep water. And this allows the blue whales to communicate with conspecifics that are far away, which is obviously beneficial for them, but it makes life a bit harder for us bioacousticians. So in order to compare the number of detections between our 4 recording locations, which are in totally different marine environments, we had to model the detection area of each recorder to consider the range over which we could hear blue whales. I'm just going to show you a map here. So this map shows the modeled detection areas of the 4 recording locations that we used. And this is for Antarctic blue whales, which produce calls at approximately 189 decibels, which is super loud. So as you can see, the detection areas of the East Coast recorders, so Kaikoura and Wairarapa, are very big, up to 250 kilometers in radius, and that's due to the deep water environment to the east of New Zealand. Conversely, in Cook Strait and the South Taranaki Bight, the listing areas were much smaller, and that's due to the land masses and the fact that the sound doesn't propagate so well in shallow water. So the detection areas for the pygmy blue whales showed a similar trend, i.e. the East Coast had larger detection areas, but overall the detection areas were smaller, and that's because pygmy blue whale calls are 6 decibels quieter, so approximately 40% quieter at 183 decibels compared to the Antarctic blue whale calls. So bearing in mind what we've just learned about the detection areas, what have we learned about the blue whales? So we're going to start with some results about the Antarctic blue whales. So what we're looking at here is the total number of Antarctic blue whale calls in the subsample data per day. And what I've done is I've scaled it per 1,000 kilometers of detection area. So basically the number of calls I detected was scaled by the size of the detection areas that I just showed you. And the data here are from the 4 recording locations plotted as colored lines. And the gray block that you can see in the middle, so end of December, January and February 2017, no data was recorded because we had the instruments out of the water. The green block that you can see is when there's no data from Wairarapa, and that was because there was a seismic survey conducted during this time, so it wasn't possible for me to go through and be sure that I was detecting all of the blue whale calls. And I should add that the Cook Strait line, so the purple line, that was only sampled in 2016. So what can we see here? We'll start off by looking at the temporal trends that we can see. So with the Antarctic blue whales, they were mostly detected during winter, so June, July and August in both years. And there was actually a secondary peak in detections during October 2016. So we've got 2 peaks and then no detections between November 2016 and late May of 2017. So the ecological conclusions that we can draw from these results suggest that actually the Antarctic blue whales that pass through New Zealand waters are highly migratory. So the winter peak in song detections represents a northbound migration of animals passing New Zealand from Antarctic feeding grounds going to warmer water breeding grounds. And the October peak are the animals that are going back again. So they're coming from their breeding grounds, passing New Zealand and going back to their feeding areas. And that's probably why we're not detecting any Antarctic blue whales in outside of winter and spring, because all the animals are in New Zealand feeding on krill. So it's not certain where the breeding areas of Antarctic blue whales are in the Western South Pacific, but as you can see, some detections were actually made in New Zealand waters during the breeding season, which I've highlighted with a little heart in September. So these song calls might have been from late animals migrating north, from early animals migrating south, or actually even from animals that were breeding in New Zealand waters. And if Antarctic blue whales do breed in New Zealand waters, that would be super exciting and have very important conservation implications. So it'd be great to look at that in the future. So that was an analysis of the temporal distributions, but now let's have a quick look at the fine-scale spatial distributions of the blue whales. So what you can't see from this figure is that actually the absolute numbers of calls were greatest at Wairarapa and Kaikoura, but because of the huge detection areas of these locations, when we scaled them by the detection area, the density of calls was much reduced. So if we focus on the winter peaks that we had, we can see that the call density is greatest in Cook Strait and the South Taranaki Bight. So just have a little map as a reminder. So while it appears that some Antarctic blue whales migrate north along the east coast, so past Kaikoura and then past Wairarapa, carrying along the east coast, there's actually a greater density of calls recorded in Cook Strait and the South Taranaki Bight, suggesting that a lot of animals use that route. So quickly moving on to the pygmy blue whales. So compared to the Antarctic blue whales, pygmy blue whale song calls were much more prevalent in the data, and they were present throughout both recording periods. The song calls were detected with highest density in the South Taranaki Bight, and especially in March to May of 2017, which is austral autumn. Pygmy blue whales have been widely reported in the South Taranaki Bight, so that was not super surprising. And it's also a known feeding area for blue whales. However, during the austral autumn, song calls also peaked in Kaikoura, and that was kind of a novel thing to find, and it'd be worth investigating whether pygmy blue whales are abundant off the coast of Kaikoura during autumn of every year, and if so, what are they even doing there? Because we didn't really expect to find them there. So, what have we learned overall? So, the acoustic presence of Antarctic blue whales was highly seasonal, so they were present during winter and spring, but pygmy blue whales were present in New Zealand waters year-round. And now that we know this, blue whales that are sighted in New Zealand outside of winter and spring, we can suggest that they're probably most likely to be pygmy blue whales, because we know that the Antarctic blue whales are less likely to be present at this time. Spatially, both subspecies were detected at all 4 recording locations. The Antarctic blue whale song calls had greatest density in Cook Strait and the South Taranaki Bight, and the pygmy blue whales were also centered in the South Taranaki Bight, but their calls were detected along the east coast as well. So the findings demonstrate that the South Taranaki Bight provides critical habitat for both subspecies of blue whale, and actually there could be some potential for resource competition between Antarctic and pygmy blue whales in the South Taranaki Bight, given that they're in the same place at the same time, and it's a known feeding area, so that's something of interest. And there's also a possibility that Antarctic blue whales might breed in New Zealand waters, which would be super exciting. I would love to see someone present that in not too distant future. So that was a super speedy run through. I'm sorry I didn't have time to go into everything. The propagation modeling is really interesting, but it's a bit dull in places, so I don't want to spend too much time on that. But yeah, so all of this wouldn't have been possible without the sponsor of my PhD and the acoustic recording project itself. So that's University of Auckland, Woodside Energy, OMV Chevron, and Marlborough District Council. And I'd also like to thank the acoustic deployment team and the colleagues at NIWA, and as well as the JASCA collaborators. Obviously, thank you all for listening. I'd be happy to take any questions.
Awesome, fantastic. So I have one question for you from the audience. Oops. Thank you. Sure. Why was the highest density of pygmy whales found around the South Taranaki Bight? Do you have any acoustic data for krill to support the idea that blue whales are feeding in New Zealand? Yeah, so I guess one of the problems with acoustic data is we know that the animal is there. We don't know how many animals there are. We don't know what they're doing. We just know that there is at least one animal there and it's making a noise. So yeah, that's kind of the next part of the project, I guess, figure out how many there are and also what they're doing when they're there. But yeah, the South Taranaki Bight is really well known is because it has an upwelling, a seasonal upwelling, which is really highly productive. So it's definitely a known feeding ground. There's been a lot of work on that from Lee Torres and Dawn Barlow. So they've got some really cool papers out about that. They studied the link between the oceanography and the krill. So, yeah, it's definitely known for feeding. Awesome, thank you very much. And for now we'll have another one, sorry. It is surprising to see the oil company amongst your sponsors. Why did you decide to take that? Oh, that's interesting. Yeah, so the acoustic project was set up kind of before I started. So obviously it's really cool that they're interested in knowing more about the wildlife in the areas that they're working. And then Woodside Energy chose to sponsor the student that was taking on the project. And they basically gave me a really nice scholarship, and I was able to do a lot of extra research that I wouldn't have been able to do otherwise. Yeah, so it was really nice for them to sponsor and to show their interest. And obviously as part of the contract, they can't change what I'm writing. They're just keen to know what is there. And yeah, so I think it's a nice way for the student to benefit, but also to benefit the company and also to benefit the conservation. Okay, one last question for now. Do you know if there are any plans to do more acoustic monitoring further north along the west coast of North Island? I'm not sure what the future plans are. I'm not sure what NIWA is planning to do with our instruments at the moment, but yeah, that would be really cool. There's a lot of Hector's dolphins up there, so if we can get the dolphins even, that'd be awesome. But obviously they're way more high frequency than the blue whales, which are super low frequency. So different hardware needed for that. Well, that's giving me a perfect link to our next speaker. Thank you very much, Victoria. So our next speaker is Max Harvey. Max is a master's student from the University of Otago. And Max has a passion for the conservation of New Zealand's threatened species, particularly marine mammals and seabirds. And today he will give us an insight into his master's thesis, which will provide guidance for the management of a small population of endangered Hector's dolphins. Hey, Max.
How's it going? Okay, I'll get the screen share up. Cool. All good? Sweet. Kia ora, everyone. Thanks for tuning in. As I was introduced as, my name is Max Harvey. I'm a master's student in the Marine Science Department at the University of Otago in Dunedin. Today I'm going to talk about some of the work done to date on my master's thesis, which is investigating the conservation biology of Hector's dolphins in Porpoise Bay in the Catlins. More specifically, during this talk, I'm going to briefly run through the preliminary results of my chapter on the effects of tourism on Hector's dolphins, and then compare this to previous research. So this photo in the background here was taken in Porpoise Bay, which is a small shallow sandy bay located on the southeast corner of New Zealand's South Island. Here, a semi-resident population of approximately 53 dolphins can be found consistently over the austral summer months. The population is particularly important, both culturally as a Tonga species, as well as a genetic stepping stone between the distinct South Coast and East Coast Hector's dolphins populations. So in a broader sense, this interstitial population may be crucial for maintaining the adaptive potential of Hector's dolphins in a rapidly changing marine environment. In Porpoise Bay itself, it's actually pretty unique. It's one of, if not the only place in New Zealand where Hector's dolphins come so close to shore without being enticed. Within the bay, dolphins can usually be found consistently between Cook Creek and South Head in the southwest corner, which is typically the calmest part of the bay. At the same time, this area appears to be favoured by swimmers during the summer, so there's potential that interactions could occur between dolphins and swimmers. And we know that from other studies in the literature that swim with dolphin interactions can potentially alter dolphin distribution as well as their behaviour, which may have cumulative impacts on the population over the long term. Therefore, during summer 2019-2020, we investigated the behavioural responses of Hector's dolphins to swimmers in Porpoise Bay in order to provide that guidance to the Department of Conservation, who are responsible for managing the dolphins. So the key aims for this research were to investigate the behavioural responses, which included human-dolphin interaction successes, group cohesion, directional responses to swimmers, and behavioural budgets, and then compare these to previous research by Bader et al. 1999 and Green 2003. So in terms of methods, our fieldwork was conducted between December 2019 and March 2020 from a theodolite station situated on South Head, down the bottom corner here, overlooking Porpoise Bay. So a theodolite is a surveyor's instrument that uses measurements of horizontal and vertical angles, as well as known parameters about the theodolite station, such as elevation, to compute geographic locations or fixes in space. So this approach allowed us to non-invasively follow dolphin groups that were using Porpoise Bay, which is really important so it doesn't affect their behavior that we were looking at. So fixes were recorded approximately every 60 to 180 seconds, while the program Pythagoras was used to input data on group characteristics, such as whether or not they had calves and group size and whatnot. So consecutive fixes form tracks from which distances and angles between dolphins and humans can be interpolated and used to analyze dolphin behavioral responses to humans. Also, while we were tracking, we made the distinction between control situations and human present situations based on definitions used in those previous research. The latter being when humans came within 200 meters of the focal dolphin group within the last 20 minutes. So to quantify the success, in parentheses, success of human-dolphin interactions, we use these methods and definitions set out by Green and Bader. So in this third column here, where it says dolphins remain less than 200 meters for 5+ minutes, that is what we considered a non-disturbing interaction. The next column along from it, we considered potentially disturbing. So when dolphins left the 200-meter radius within a 5-minute time period. And finally, we considered interactions disturbing based on whether they left the 200-meter radius of swimmers within 2 minutes. So in total, 109 human-dolphin interactions were considered for analysis in the study. In 86% of these tracks, dolphins remained within 200 meters of swimmers for at least 5 minutes, so we're considered non-disturbing. And 8% of the time, dolphins left the 200-meter radius within 5 minutes, and then 6% of the time, dolphins left the radius within 2 minutes. So it appears that most of the time, human-dolphin interactions, human interactions were not displacing the dolphins in the short term, at least. It's noteworthy, however, that unaided swimmers were the most observed swimmer type during the interactions. And out of all the swimmer types, unaided swimmers had the greatest incidence of potentially disturbing interactions, with 7 out of the 15. So this could possibly have implications for management, where we target this swimmer type or the swimmer group with management strategies that reduce potential disturbance by educating them on the potential impacts of swim with attempts. Compared to previous research, we found that, interestingly, the proportion of time that dolphins spent in human presence has increased quite dramatically since the first study in 1997. So it's up to 55% of the observation time dolphins were in human presence. But at the same time, the proportion of non-disturbing tracks has also increased quite a bit as well. So it's gone from 57% in 1995 to 97 to 77%, and now 86% of interactions are considered non-disturbing. So this study, we can't really conclude why these trends exist, but we can kind of speculate as to why there's a greater number or greater proportion of non-disturbing interactions. One may be that the guidelines set out at Porpoise Bay, meaning that there's less swimmers contradicting them, and so less potential for dolphins to be disturbed. Alternatively, it could be that Hector's dolphins in Porpoise Bay have become habituated to swimmers over the long term due to exposure over the last 25 years. However, we can't conclude the exact cause of this trend. Another key component in investigating the effects of tourism in this study was the assessment of human impact on Hector's dolphin group cohesion. So in other species, altered group dispersal or group cohesion has been shown to have effects on individuals as well as potentially long-term effects on populations. So in this study, we recorded dolphin group cohesion with each fix, where tight states were considered, tight states were recorded as when the average distance between dolphins in the focal group was between 0 and 5 body lengths, and dispersed states being when the average distance between dolphins in the focal group was between 5 and 10 body lengths. So what we found was that dolphins were more likely to be in tight states during control situations when there's no people around, and the opposite's true when people were around. Dolphins were more likely to be in dispersed states. So this is kind of interesting. It's the opposite to what Lars Bader found in 1995 to 97, where dolphins became tighter in impact situations around swimmers, but as well as around boats, because back in those first two studies, there also used to be a tour boat that operated in Porpoise Bay, but it's no longer there. It's just sort of shore-based tourism that's the potential impact here. But our results here are more in line with what Aaron Green found in 2003, where groups became more dispersed in the presence of humans. We also investigated how dolphin orientations changed in response to different explanatory variables, such as whether or not calves were present in the group, the time of day, the month, and the time into an encounter. So this diagram on the left here shows how the program Pythagoras classified dolphin headings. So between two consecutive points, say this dolphin's going between point A and point B, it is considered going towards the swimmer. So the swimmer being at 0 degrees here and the dolphin being at center. So based on this way that we've classified the headings, we'd expect that 0.25%, so 0.25 of the time, dolphins would be going towards swimmers. So we used generalized linear mix models to assess the probability that dolphins heading towards swimmers. This was chosen because we had repeated measures on the same dolphin group, so they could be tracked in relation to multiple swimmer groups. So we included that random effect of group ID here. So the candidate models on the right here in this table were ranked due by AIC score, which takes into account model fit as well as complexity. And we found that the best fitting model was the null model. but there was some support for the other models as they fall within 2 AIC points of each other. But considering this null model, which excludes 0.25, we would say that dolphins are significantly attracted to swimmers in this case. So we also looked at, where are we going? But we also looked at, in order to compare to previous research, the effect of time into an encounter on the probability that dolphins head towards swimmers. We found that up until the 50th minute, dolphins were significantly attracted towards swimmers, after which, because it excludes the 0.25, the confidence interval is outside 0.25, after which the 50th minute, they were no more attracted to people than would be considered if their movement was random. Finally, we looked at the impact of human presence on dolphin behavioral budgets. So it should be noted before I get into this that behavioral observations of cetaceans can come with a caveat, especially from land, because we can't reliably know what they're doing under the water most of the time. But these behavioral budgets can give us an idea about potential changes to the animal's behavior. So we found that in the presence of swimmers, dolphins reduced their diving, the proportion of time that they were diving, quite significantly. I should note as well that I haven't carried out any statistical analysis on this yet. We're still figuring out the right way to conduct that. But in control situations, dolphins were more likely to be diving and what we might think of diving as potentially feeding, whereas when humans were present, they were more likely to be milling. Again, this is in line with what Erin Green found in 2003 in her study. So that's just interesting to note. We're not sure why it's similar. It could be that the dolphins have become habituated and are displaying the same sort of behavioral trends over time, or it could be something else. So just a quick summary of the findings so far. The results of this study suggest that most human-dolphin interactions in Porpoise Bay during summer 2019-2020 were non-disturbing to the dolphins based on the definitions used by previous research. Human presence was shown to alter dolphins' dispersal state or their cohesion, as well as their behavioral budgets, and there's evidence that suggests that dolphins in Porpoise Bay are attracted to people, not to swimmers. It's interesting to note, however, that this study, an additional part of the study, found no evidence for a change in population size of the Hector's dolphins using Porpoise Bay over the last 25 years. But it's unknown what the population, the unimpacted population size would be otherwise. But so in summary, at present, there's no evidence that tourism has impacted the population over the long term. But it's unclear how we would actually measure those population level effects, given that there's such a wide time period between previous studies. In terms of management recommendations, to continue, or we suggest that they continue to proactively manage human-dolphin interactions in Porpoise Bay. And interestingly, something that I came across the other day was that under the Marine Mammal Protection Regulations 1992, it states that people may not swim with juvenile dolphins or pods containing juveniles. Out of our tracks, so 51 out of 109 dolphin tracks, so 47% actually included calves in the study. So one suggestion to DOC that they may follow up on is adding signage or clarifying signage around swimming with juvenile dolphins. In addition to this, we just recommend that they continue to support the monitoring of the Porpoise Bay population, particularly given new fisheries regulations that were decided upon this year as a part of the threat management plan, and due to that changing tourism landscape due to COVID-19. So we're not actually sure whether or not this, this snapshot in time from this study is going to apply to the future. In terms of my own research, so we've already estimated the abundance of Hector's dolphins during this past summer. Next up, I'm going to assess the DIL use, so the daily usage patterns of Hector's dolphins in Porpoise Bay using passive acoustic monitoring, and then look at some kernel density analysis, so figuring out the preferred area of dolphins within the bay. And finally, I'd just like to give a huge thank you to all the volunteers that helped with the field work. There was between 15 and 20 of you. You know who you are. Really thanks a lot. Ros Cole from the Department of Conservation in particular, She was massive in getting this project off the ground, and it probably wouldn't have happened without her. The University of Otago for providing equipment, as well as some funding, and the Whale and Dolphin Trust for providing funding and equipment as well. Lastly, just a huge thanks to Nick, Moana and Tamatea, some locals down from the Catlins that just kept me sane while I was down there for 10 weeks over summer, and were just a massive support while I was down there. Sweet, that's it, thank you.
Fantastic, thank you very much. And we have some questions for you. What is the total population size? Is one of the questions.
So the total population size of Hector's dolphins around New Zealand, the last time it was estimated, it was around 15,000, although that's sort of contrary to the previous estimate, which was around 7,000 a few years ago. But the official abundance estimate for the whole of New Zealand is around 15,000. In Porpoise Bay itself, it's between 40 and 67, I think, 41 and 67. It's around 53 dolphins.
Fantastic. And another comment or question. First of all, nice work. And did you find that different age classes, for example, juveniles versus adults of dolphins, were more likely to interact with swimmers?
Well, thanks for the question, first and foremost. We haven't actually investigated that yet. That's sort of something on the horizon. There's just different questions popping up as I'm trawling through the data and doing these different analyses. But that is something that I'm considering looking at. I'm not sure how I'd do it at this stage, but it's tedious as well with the methodology that we used. So when we follow a dolphin group, we follow it for as long as possible before, say, conditions deteriorate or the group is lost. So sometimes with Hector's dolphins, their sort of social system is that fusion-fission. So two groups might come together and then split apart again. So to get around this, we would just follow the largest group that splits out of that. So it's possible that sometimes mother and calf pairs or something like that could be a part of that large group and then split off into a smaller group. So we didn't necessarily look at what their response was. So yeah, I haven't looked at it yet, but it's something to consider in the future.
Okay, and the final question, does group cohesiveness change during the reproductive season in response to swimmers?
Not sure. So our study only ran from December through to March. So I think we'd need a larger data set that included, the rest of the year and that to be able to fully answer that question. But unfortunately, that's out of the scope of my study in particular.
Okay, thank you very much.
Awesome.
So our next speaker is also giving us some information about Hector dolphins. It's Lindsay Wickman from the University of Otago, and Lindsay has a special interest in marine mammal conservation and population dynamics, while also taking advantage of public outreach opportunities along the way. And she's currently doing her PhD at the University of Otago, updating Hector's dolphins population status around Banks Peninsula.
Awesome. Thanks so much for that intro, Marika. And thank you so much for everyone who is here watching. All right, Max, can you see my cursor?
Yep.
On the screen? Okay, cool. Thank you. All right. Kia ora koutou. Everyone, I'm Lindsay. I'm a PhD student at the University of Otago, supervised by Steve Dawson, Liz Sleaton, and Peter Dillingham. And today I'm going to share with you some current progress during my PhD project on preliminary estimates of survival rate using a Bayesian multi-state capture-recapture model. So this is still a work in progress. So Hector's dolphins are known locally around Banks Peninsula as Tutamita kurai, which means special ocean dweller in Māori. They're endemic to New Zealand, and they are an endangered species. And their primary threat is set netting. And we know that set nets are impacting the population partly from analysis of survival rates. So survival rate is the probability that an individual survives from 1 year to the next. and we estimate survival rates using a long-term photo ID dataset. So these dolphins have been studied from 1985 to the present using the same methods throughout this entire period. So that includes a zigzag transect through the harbor and also a coastal strip transect that goes all along the entire coast of Banks Peninsula and into all of those little bays. An estimation of survival rate continues to be a really high priority for this research program because it's so important for determining whether or not the population is declining and whether or not protection measures are working and being effective. So the last time survival rates were estimated for this population was by Andrew Gormley using data from 1986 to 2005. And he found an increase in survival rate after the establishment of the Banks Peninsula Marine Mammal Sanctuary in 1988. So although these survival rates have been really key to assessing the effectiveness of this sanctuary, it's definitely time for an update. For one, there's some contention over how accurate these survival rates are, partly because it's been 15 years, so we have 15 more years of data to use. And also, they don't account for more recent protection measures, which include an extension of set-net protections north and south. So for my PhD project, one of my main objectives is to develop a new model for estimating survival rate. And we chose a Bayesian model so that we could have a lot of flexibility in building a model unique for our population. So of course, the first step in updating that population model with survival rates is to do photo ID matching. So this is a work in progress. Every year, over 3,000 photographs are taken, and then these have to be matched to over 600 highly distinctive individuals in the catalog. So far, matches have been made all the way up to 2015, and just to give you an idea of how many matches have to be completed, just between 2012 and 2015, there are about 1,000 different matches made. So this is still in progress. I am currently in the process of matching up to 2020. So each time we take a photograph of an individual and match it to our catalog, that creates a record of whether or not that individual is alive and photographed in that year. And that allows us to build a capture history. So down here is an example of a capture history. So the zeros represent years that the individual was not photographed, and a 1 means that it was encountered and photographed this year. So for each individual gets a capture history, and those capture histories are what's put into a capture-recapture model, which estimates survival rate. So when we put this into a model, the model will know that at least from this first one here to this last one at the end of the capture history, the individual was definitely alive. But then there is a 0 at the end of this capture history, and the model has to make a decision. Was that individual alive and we just didn't photograph it, or was that individual dead? So that's where capture probability comes in. And capture probability is the probability that if an individual is alive, what is the probability that we encounter and photograph it this year? So for example, this individual, because looking at its past, there's been multiple occasions where we haven't seen it in 2, maybe 3 years. It's likely this capture history is relatively low, and this 0 probably doesn't mean that it's died. It's probably still alive. So estimating capture probabilities is really, really important for estimating survival rate. but it can be a bit tricky if capture probabilities vary between individuals. And one way that capture probabilities can vary is if individuals have different home ranges. So we know from past work by Will Raymond that this is likely to be true. We know that Hector's dolphins have relatively small alongshore home ranges of just about 50 kilometers that are centered around certain parts of the peninsula, and these home ranges tend to stay relatively constant over time. So over here is a schematic that shows the home ranges of individual dolphins. Each bar is a different dolphin, and it represents their home range. So over here in the middle is Akaroa Harbor. So these two dolphins here, their home range is centered in Akaroa Harbor, which is where we survey most frequently. And then these dolphins over here on the end of the graph, their home ranges are centered in the northern part of Banks Peninsula. And as you can see, their home range actually overlaps with the edge of where our survey area actually is. And because we don't survey that area as frequently, and because they may also sometimes spend some of their time outside of the study area, the capture probabilities of these individuals over here are likely lower than the capture probability of these individuals that have a center of home range near Akaroa Harbor. So to account for these heterogeneous capture probabilities, we decided to pursue a multi-state model where the chance of resighting an individual depends on their home range and search effort. So to do this, we split the peninsula into 3 different areas, 1, 2, and 3. And in each region, capture probability is allowed to vary. So for example, we expect that dolphins with home ranges in area 2 and 3 likely have lower capture probabilities, again, because we don't survey these areas as often, and these areas border the edge of our survey area where dolphins may actually leave. And the reason that we're doing this is because if we don't, then not accounting for these lower capture probabilities can bias our estimations of survival rate low. So if we don't account for this, the model will think that an individual in zone 3 is dead when actually it's just left the survey area for a little bit and has a lower capture probability. So keep in mind, right now I just have 3 different zones around the peninsula, just for simplicity's sake while I'm developing the model, but we are going to expand it to 4. So inputting data into the model is really similar to a regular capture-recapture model, where you input a capture history for each individual. But for this multi-state model, we separate the capture histories out into 3. So for each individual has 3 capture histories, 1 for each zone. And then each individual also has a record of where that individual was last seen. So for each year, each individual gets assigned a zone, which allows the model to choose a capture probability and estimate survival rate. So we have developed the model and it is running, and luckily we're getting survival rate estimates that are looking reasonably realistic. So our median estimates of survival is about 0.901, which means that an individual has about a 90% chance of surviving from 1 year to the next, and that's comparable to past estimates of survival, like the ones I showed you from Andrew Gormley's work. But there are a few potential anomalies, like this drop in survival rates in 92 and 93. I'm not sure if that's real and it needs to be investigated. So we're still error checking the data in the model. So again, these survival rates are preliminary. And of course, the next step is to include all the years of our data set, so all the way up to 2015. And once the photo ID matching is completed, we will add that data as well. increasing the number of zones from 3 to 4. And then we'll also want to compare this model to competing models, for example, one in which there's a constant capture probability between zones rather than allowing the capture probabilities to vary. Then hopefully we'll be able to answer questions like what the trend in survival is over time. And in addition to allowing capture probabilities to vary over zone, we can also allow survival rate to vary between zones to see if there's a difference in survival rate between areas. So just to summarize, again, this multi-state capture-recapture model is a work in progress, but the framework does provide a way of dealing with the heterogeneous capture probabilities in our population. Preliminary estimates are similar to the estimates of survival rate from Gormley, but we need to continue checking the model and data set and add more years before we can make any conclusions. So I'd just like to quickly thank the Department of Conservation for funding this work, and the New Zealand Whale and Dolphin Trust and the University of Otago, my supervisors, and also the University of Otago Marine Mammal Lab, and all the past students and volunteers that spent hours and hours and hours collecting all of this photo ID data to put into capture-recapture models. Thank you.
Awesome. Thank you very much, Lindsay. I have a question for you from the audience. Do you conduct surveys multiple times throughout the year? And if so, have you considered using the multi-stage open robust design model rather than assigning only one area per individual over a year?
Sorry, could you repeat the last part of that question?
Yes. If you considered using the multi-stage open robust design model.
Right. Yeah.
Rather
So
than assigning only one area per individual over a year.
So the first part of that question, how many surveys do we do? It depends with each year, but we do multiple surveys throughout the year. The surveys are concentrated between the summer months of December and March, and we go out and do a survey anytime we can, depending on the weather. And we choose which area to survey based on the best weather conditions. So there are many different, many different survey days per year. As far as the robust design, I still actually need to look into that a bit more. Thank you so much for the feedback. I'll be looking into that, and I'll get a better answer as to, but we'll definitely be looking into that as a potential competing model.
Okay, one more question. How stable is the home range of Hector's dolphins through the time? Does it change during the reproductive period?
So there's no evidence that it changes through the reproductive period. The work that Will did on home range was using data from 1986 to 2009, and that showed that the home range was really, really stable, but we have not looked at home range since then. I'd expect that adding more data, we might find that home range is actually a little bit more variable, but again, that work just hasn't been done from the evidence that we have. And then, is that a question from you?
No.
Okay, sorry. I thought Max was asking me something. Good.
Okay, thank you very much, Lindsay. Okay, now let me introduce you our last speaker, which is Dr. Krista Hupman. Krista has been researching leopard seals in New Zealand waters for the past 6 years. She currently works as a marine mammal biologist at the National Institute of Water and Atmospheric Research and is the co-founder of the nonprofit organization leopardseals.org. So make sure to have a look at this webpage. Today, she will give us a global review of leopard seal birth, pups, and records. Birth and pup records, sorry. It's your stage.
Thank you, everybody. Just organizing my sharing of screen and should be able to see there. Okay, great. Thank you very much for having me today as part of this seminar series, and thank you to everybody who's watching as well. Thank you for the introduction, and as they were saying, I will be speaking today about a global review of leopard seal birth and pup records, and including a few records from New Zealand. And I'd like to start off by thanking my co-authors, Dr. Ingrid Visser, Rick Bout and Martin Cawthorne, and thanking the organisations involved in this study, which is NIWA, leopardseals.org and Otago Museum. So to start off, leopard seals distribution. So leopard seals are found in the southern circumpolar regions and also in the northern hemisphere. But the majority of their population is distributed south of the subantarctic islands and in the Antarctic. And they're known to primarily inhabit the pack ice, but they do have higher densities that occur southwards towards the edge of the continent as well. And when we look at a global kind of range for leopard seals, we can see that during the austral spring and summer, leopard seals seem to travel southwards. However, during the austral autumn and winter, some leopard seals will also travel northwards. So this is a normal movement pattern which occurs. And we've actually documented their presence. Obviously, we know of leopard seals mainly being around the Antarctic, and that's due to the presence of sea ice. But we actually do get a number of individuals which are found around the subantarctic islands, also within South America, South Africa, Australia, New Zealand, and we even have some records in the tropics, which is quite unusual. But as these records become more and more, we're finding more and more northern records. When we look at leopard seals in New Zealand waters, they've been present for some time. So they've been present back in the 12th century. There's over 3,750 sightings from past to present. They're recorded all year round. So before we thought that they were just seasonal, but they're not. And they are in all regions of New Zealand, and they do include immature and mature individuals of both sexes. So this is sort of knowledge that we've learnt over the last 5 years of studying these animals just via citizen science records. And when we look at their reproductive biology, they're actually a solitary species, and there's very limited knowledge on their reproductive history. We do know that they're sexually mature around 2 to 7 years old, and they only give birth to a single pup. And when we actually look at leopard seal births, there's not a lot known. They are presumed to give birth on the pack ice surrounding Antarctica. That's how we would normally think of leopard seals. And it's presumed that they'd normally occur during the austral spring and early summer. But there's actually a few difficulties when we're actually recording leopard seal births and pups. So the first is there's actually no definition of what is a pup versus a birth. So that can make record quite confusing. There's also no database anywhere worldwide that we found that collated birth and pup records, and therefore that was the initiation of this study. So the objectives were to basically develop some type of standardized system to be able to log what is births and pups, to be able to present a global review of actually what records exist out there, and lastly present New Zealand births, which is sort of where we started at the beginning and found out that there was this sort of gap in the literature that we wanted to fill. So just running quickly, through the methodology, we reviewed all the published literature to find out what was available. We then reviewed the doc database to see if there were any birth records in here. We completed any field observations of births that occurred. Only one did, but still were some. And we developed that standardized system to be able to determine what constitutes a birth versus a pup record. And so this is just an outline of that. So basically the first thing was we looked at specific features which were determined for births. So that was photographic video evidence or descriptions of any type of leopard seals actually being born, or any animals with a fresh umbilical cord or having a placenta. So we classified them as a birth, whereas we didn't have any specific features that were only for pups. For the age, we looked at animals that were less than 48 hours, 2 days old was being a birth, whereas greater than 48 hours old, 2 days old was a pup. We looked at the standard body length of less than 160 centimeters was a birth, whereas 161 to 220 was a pup. And any time that a description was used by an author, which was actually relating to a leopard seal birth or a pup record, we actually classified those as probable births or pups. But for the actual records here, we'll be only giving you the ones that were certain, not probable. So moving on to regions, we actually classified leopard seal birth and pup records into regions of Australia, New Zealand, the subantarctic islands, the Antarctic, Falkland Islands, South America and South Africa. And we actually looked at a few attributes for each of these records, which was what month they were born or pupped in, the region and month together, what substrate they were born on, if there was a presence of a mother or not, and a presence of something called lanugo, which is actually a neonatal fur, which is an indicator that it is a birth or a pup record. So to give you an idea of our results, we had 15 birth records in total and 43 pup records. And when we actually analyse these by month, you can see that they are spread over sort of that September to March period, which is quite different, I guess, to what we found in the published literature for births alone. It was sort of saying that they were only in sort of those November, December, maybe early January months, but we've actually found that they're spread across a lot wider months. And pups, there wasn't a lot sort of written about the, you know, the normal pupping period, but we can actually see that that spreads quite wide all the way from September till March. When we looked at records by region, it was interesting to see that for birth records, there weren't many in the Antarctic, so there's only 4 birth records that we have in the Antarctic. However, when we look at New Zealand, for instance, there's 3. So it actually shows that even though we know that the Antarctic is the stronghold for this population and sort of the main birthing region, there really aren't a lot of birth records. However, where that really changed was the pup records. So there were a number, sort of 33 in the Antarctic and actually none of those in New Zealand. So it's interesting to highlight the differences between the two. When we're looking at births by the region and month, and you can actually see that sort of there were some earlier records in sort of New Zealand and the Falkland Islands, subantarctic islands, and that's, you know, they move later in the months when it comes to somewhere like Antarctica and the Falkland Islands. And same with pups, you can see that there aren't sort of many pups in those original early months. However, you start to see those pups in Antarctica sort of around the November, December, where you do have still some pups in March in Antarctica, so they're quite spread out. Substrate is an interesting topic. So basically we would have thought previously that all leopard seal births were on ice, and same with pup records. However, we show that that's not necessarily always the case. So there was a quite horrific study done back in the day where a leopard seal was caged for its pregnancy, and basically they kept it in that cage until it had given birth, so they could watch when it gave birth. So that was the cage record. We had one on grass, and then we had some interesting ones on a boulder beach and a sandy beach, but very interesting, in New Zealand we had one leopard seal born in the water. That's the only record that we've ever had born in the water, so that was interesting. The mother was present in 9 out of the 15 birth records and 24 out of the 43 pup records. And when we looked at the presence of that lanugo, that neonatal fur, it was present in 8 of the 15 birth records and 6 of the 43 pup records. So just moving on quickly to the New Zealand births, the first birth that we have recorded was in the Chatham Islands back in 1972. And there's not a lot of information about this other than the pup was born on the Chatham Islands and it died shortly after birth. The second one was on the Curvia Island back in 1977. The only information we have here was that it was observed giving birth and the pup appeared to be in good health, but it actually died the next day. You can see some photographs we actually chased up of that old record, which was great. Now, Dunedin was a really interesting birth. It was an animal which was born in October, and I was very happy to be able to go down and actually see this animal after it was born. So I arrived there on the night that it was born, and just to give you a bit of a brief outline of what happened, we first had a mother which was observed in the shallow water, and then a local surfer actually found the leopard seal pup sort of on its own in the shallows, and it looked like it was drowning. So he actually picked it up, and there's just a little disclaimer here saying, please never get this close to a leopard seal. It's just that this was a very extenuating circumstance. So the surfer actually picked that leopard seal up and popped it next to the mother. But unfortunately, the mother really didn't have a bar of the leopard seal. It wouldn't allow it to suckle, and it kept moving away from the animal. So the animal was left alone, and basically the mother swam away, and it wasn't until the next day Department of Conservation came in and decided that the best case was to euthanize the animal because it wasn't suckling. So just as a final conclusion, one of the things that's important about these records is to highlight the difference between somewhere like Antarctica and New Zealand. We think for the importance of populations that Antarctica is a stronghold and New Zealand is a northern habitat. And when we look at population size, it's very large in the Antarctic, around 35,000, whereas we really don't even know the number here for New Zealand, maybe around 100 individuals we have ID'd. And while Antarctica is considered a core breeding area and New Zealand is a limited breeding area, we actually have only 4 records of births in the Antarctic and 3 records in New Zealand. And actually the spread of records in New Zealand is better than what we have for the Antarctic. So that was quite an interesting record. Also looking at the birthing and pupping period, the presumed peak for birthing was between November and December. But when you actually looked at the actual peak from our records, it showed that that was a lot earlier, around September to November. And the actual peak in pupping was actually a lot later, so sort of November, December and January. So what we actually took from this is that classifying those births and pups by a standard definition is really important, because actually the presumed peak birthing period turns out to be the actual peak birthing and pupping period together. So that's a really interesting finding. And just lastly, a couple of points to say that systematic sightings should always be collected throughout these animals' range. We really have a need to improve our understanding of their reproductive ecology. And obviously it's important to conserve these species because we are in an ever-changing global environment and we don't really know how these sightings coming north are going to fare in time to come. So just lastly, I'd like to thank the New Zealand Marine Sciences Society for having me, all my co-authors, NIWA for supporting this work, all the volunteers and researchers of NIWA and leopardseals.org, Givney Forbes for assisting in the field, Jim Fife for alerting us from Department of Conservation about the birth in Dunedin, Tony Sawyers for alerting us to one of those pup sightings, Otago Museum for all of their help with the pup when it was down in Otago, Dalton Williams for actually finding that leopard seal pup, and everyone who contributed sightings to the database. And just to leave with one slide, if anyone sights a leopard seal around New Zealand, we'd love to hear from you on any of these channels. Thank you very much.
Thank you, Krista, for this great talk and the beautiful images going along with them. I have a question from the audience.
Sure.
Do you have a breakdown of all the births where the pup died soon after? Seeing as in New Zealand, all 3 have died, it would be interesting to know whether this is normal in other places away from Antarctica.
Yeah, it's a really good point, actually. So there's a very limited information on these births and pups, especially the births. The births that we do have recorded in northern regions, it seems that none of them are viable at this stage, but that is something that we're currently writing a paper on, so we hope to put out a little bit more information. But yeah, it's very interesting to note that, you know, these animals are somewhat breeding north because they are actually having babies, but if they're not viable, then is that constituting breeding? It's a very interesting question.
Okay, one more question. Great talk first. And curious, how was the water birth recorded? By chance and by who?
Yeah, very interesting. So Mr. Dalton Williams, he's a young surfer down in Otago. He was literally coming out of the surf and he noticed the leopard seal sort of struggling around in the water on its own. And then he noticed a mother that was actually on the beach. So he basically picked it up just out of natural instinct. Normally we would say, you know, never interfere with a mother and a pup because then it can sometimes abandon it. But this animal was clearly drowning. And so he felt that there was nothing else to do. So he definitely did the right thing. And yeah, he just basically pulled it up onto the beach. Called Department of Conservation, and Jim Fife, a local ranger down there, let us know, and we got on the plane and we got there as soon as we could. And yeah, really unusual circumstances and real miracle to be able to see this animal. And then we actually were able to do a necropsy on the animal, and it's now displayed at Otago Museum, which will sort of be a part of our next of publishing about this animal. But yeah, it's really interesting to be able to have a record like that with so much information now.
Yeah, that's true. Thank you very much to you and to all our speakers today, and for all the people in the audience joining our webinar today. Make sure to check out our website of the society, nzmss.org, if you need information about our society or want to become a member as well, which is actually really cheap, especially for students. So thank you everybody for joining today, and see you next month on the 15th of October for this webinar with the topic Mataranga Moray in our Moana. Have a good evening. Bye.